Data submission. Let's start with the data submission. In general, the data submission to INDA is as per the formats accepted by the International Nucleotide Sequence Data Consortium or INSDC. Different type of nucleotide datasets need to be appended with suitable metadata to ensure discoverability and appropriate interpretation. Thus, all submissions must include various data metadata groups such as study, sample, experiment, run, assembly, and annotated sequences. A study is defined as an entity that helps in grouping the different types of data submitted to the data archive and controls its date of release. The sample has information regarding the sequenced source of the material. An experiment is an object which contains all the information regarding a sequencing experiment, while run is the actual NGS raw data file. IBDC and INSDC accessions will be simultaneously provided for the study, sample, experiment, run, assembly, and annotated sequence submissions. For NGS data submission, the main components to be registered are study, sample, and experiment. Users can upload raw data generated from transcriptomic and genomic experiments in FASTQ, BAM, or in CRAM file format. For assembly submission, study, sample, experiment, and assembly files have to be submitted. Assembly using already submitted INSDC accessions for raw data can also be submitted. Contig, scaffold, and chromosome-level assemblies can be submitted. The file types in the assembly submission are FASTA file, AGP file, GFF3, chromosome list, and EMBL flat file. A study, the DNA sequence in FASTA format, and the relevant annotations need to be submitted. For annotated DNA sequence submission like rRNAs, mRNAs, ncRNAs, and many other types of sequences.